required. So I'm assuming you can hear me and see my screen. Meanwhile, I request you to let somebody in the meeting if somebody tries to join when the presentation is on. So hello, this is the NPTEL Cell Biology Cellular Organization, Division and Processes. We'll have these sessions every week. That is meant to be a discussion session that we will share extra information on the course content, discuss papers, or discuss previous year assignment questions. The course instructor for this course is Professor Shika Laloria from IIT Bangalore, and I am one of the PMRF TAs. Such discussion sessions will happen every Saturday, Monday, and Tuesday, and you can join any one of them based on your schedule. So last week, when we discussed as to what should be discussed in these sessions, most of you said that they want to hear recent papers or discoveries or techniques that are used in the topics covered, and that is what I'll try to cover today. But I will also be discussing the previous year assignment questions that we can solve together. So to begin with, to give you an overview of what happened in this week's course, we saw what is the regulatory factors that control cell cycle in a cell. This was first identified in yeast, and later extensive research has now helped us identify such processes in vertebrates and higher mammalian cells as well. So we see that the cell cycle is typically divided into 4 phases: the G1 phase, the S phase, the G2 phase, and the M phase. In the G1 phase, the cell is slowly preparing for DNA replication. It is making the necessary proteins and that is required in DNA replication, and it is making the cell ready. Then after it is committed towards cell division, there is the replicative phase, wherein if your cell already has 2N of DNA content, it doubles and becomes 4N of DNA content. Then is the G2 phase, wherein the cell prepares for cell division, typically grows in size, but there might be certain cell divisions such as those seen in embryonic cells, wherein there is no growth phase, but typically there is a growth phase. And finally, the M phase, which divides the mother cell into 2 daughter cells. The M phase is what we've seen and more familiar with going through these mitotic stages, wherein the chromatin is condensed into chromosomes, it assembles at the center of the cell, and then it is pulled towards 2 poles. And then finally, there is a cytoplasmic division, which results in 2 daughter cells. Now, here we need to appreciate the fact that after dividing, the cell can attain this G0, which is like a phase in which the cell is not actively dividing. Such non-dividing cells can either be senescent, which means that they've halted their replication because of some stress. Say the DNA is not replicated properly, the cell have some other infection or something which is forcing the cell to not divide. This is irreversible. These cells will never divide again. But there are also post-mitotic cells that might be terminally differentiated, but there might be certain stimulation that can be given to such post-mitotic cells, and they can re-enter the cell cycle. So this regulation needs to be precise, because we know that uncontrolled cell division often leads to tumor formation or cancer, and hence there are various factors that control this process. To simplify it, we can say that there are cyclins that are degraded at the end of every stage. So it comes up and goes down, the levels comes up and goes down based on what cycle of cell division it is in, what stage of cell division it is in. And then there are these cyclin-dependent kinases, CDKs, that are present throughout the cell cycle. However, only when a cyclin and CDK form a complex, then the complex becomes active. There is phosphorylation of various target proteins, because of which the cell cycle advances. Now, this is further controlled by other proteins such as that are either inhibitory in nature or activatory in nature. If there are inhibitory proteins, then they will phosphorylate or dephosphorylate certain essential residues, which makes this complex non-functional. At the same time, activators are those that can again typically dephosphorylate or phosphorylate crucial residues that makes this complex active. We have seen a few examples through this content of this week in the course. Now, again, research basically using mutation studies wherein they constructed mutants that were unable to cross a particular stage of cell division and identified proteins that were crucial for this process. And this led to the current knowledge that we have, wherein in vertebrate cells, there are 4 major cyclins, D, E, A, and B, that go through the cyclic stages of upregulation and downregulation. Similar homologs can be found in yeast and other lower organisms, and they are named as CLN3, CLN1, 2, 5, 6, and so on. The CDKs were initially identified in the yeast, and they were known as the CDC28 or CDC2 in fission yeast or CDC28 in budding yeast, and that is now known in vertebrates and higher organisms as CDKs. While in yeast, if you can observe that the CDKs are the same in different stages of cell division, in vertebrates, there are different CDKs that bind to these different cyclins and have unique and more complicated regulation of cell division. So there was this very interesting discovery that was made by these 3 scientists, and they were awarded the Nobel Prize in Physiology and Medicine in 2001 for this discovery, wherein they said that it is not sufficient if a cyclin and CDK complex is present. There are also these checkpoints that are present in these cells. Checkpoints basically are other proteins that control the activity of cyclin and CDKs, and they can be inhibitory or activatory in nature. Most often than not, they are inhibitory in nature, and there are 4 such important checkpoints. One happens in the late G phase, and this is often known as the start checkpoint or the replication initiation point, wherein the cell is committed towards cell cycle. Here, the checkpoint is basically for the availability of nutrients, availability of growth factor, whether or not there is DNA damage. If the cell divides, will there be healthy daughter cells produced or not? And then is the G2 checkpoint, which comes after DNA replication. Here, it basically checks if the DNA replication is complete, if there is any DNA damage, or if the cell is of sufficient size so that it can undergo cytokinesis. Next is the mitotic checkpoint, which is one of the most crucial checkpoints because here it checks whether there is a proper attachment of the kinetochores to the microtubules. Now, if these chromosomes do not attach to the microtubules, then there is improper segregation. One cell can get more chromosomes, one cell can get lesser chromosomes, and this is known as aneuploidy and results in catastrophic and often fatal results. Then finally, there is a cytokinesis checkpoint, which controls the cleavage of the cytoplasm into 2. Now, this is an oversimplified version of the cell cycle. However, there are thousands of proteins and molecules that play very simple and very crucial roles in regulating this process. Now, moving on, let's just look at the 10 questions that were the previous year assignment question for the same week in the NPTEL course, so that we can try and solve this together and get an idea as to what the content, if we've understood the content properly or not. So the first question was, according to the experiment conducted by Johnson and Rao in 1970, what would happen when the G1 and S phase nuclei are fused together? Now, this was an experiment that was conducted when nobody knew if cell cycle was in fact regulated or not. They just saw under the microscope that there was a cell that was dividing into 2, and they had no idea as to how this is happening, whether this is regulated or is it a random process. So what they did was they fused 2 cells in different stages of cell cycle. And they saw that when an S phase cell is fused with that of that cell in the G1 phase, then the G1 nucleus also starts dividing, also starts replicating its DNA and enters S phase. However, the same was not observed when an S phase nucleus was added to a G2 phase cell, or when a G1 phase cell was fused with that of a G2 phase cell. This suggested that there is something in the S phase cell that is able to trigger the division of DNA in G1 as well. Now we know that this is the mitotic promoting factor, MPFs, and what basically happens is, in the S phase there are these in the S phase there are these S phase cyclins which will degrade or help in advancing of the cell cycle, and the G1 phase will halt and the cells move to S phase. However, because in SG2 or G1 and G2, these are not consecutive stages in the cell cycle, they do not influence each other, suggesting that cell cycle is not only regulated, it is also only possible in a sequential manner. The cell will go through these stages only in the same sequence. So the answer is that the G1 phase cells enter S phase prematurely. The second question was, which of the following statement is correct regarding V1? It is an inhibitory kinase that phosphorylates CDC2P, or if it's a activating kinase that phosphorylates CDC2P, and varying residues that they have mentioned here. So if you can recall that in the course content, the course instructor mentioned that V1 was a mutant that was observed that once this gene is mutated from the cell, the cells do not grow in size and they do not have this checkpoint for the cell size. And because of that, the cells that are formed are very small in size and there's very fast cell division happening. So what it actually does is that it's an inhibitory kinase. It inhibits the M cyclin complex with the CDK. So if V1 is present, even though this complex is there, this complex is phosphorylated at an inhibitory residue, and because of this, it cannot go and perform its downstream activities, because of which the cell is halted. Only when there is the CDC25, which is a phosphatase, only when this removes the inhibitory phosphate mark on the CDK cyclin complex is this active, and the cell can proceed with mitosis. So this acts as a checkpoint wherein what happens is, if the cell has not attained their proper size, then the CDC25 is inhibited, because of which this V1 will transfer the inhibitory phosphate group. However, there will be no CDC25 to remove it. Hence, the cell cycle will be halted up until it reaches a point where the cell decides, okay, so the right size, CDC25 is activated, and because of which the inhibitory phosphate is removed. So the answer is that V1 is in fact an inhibitory kinase that was obvious, and this phosphorylates CDC2, which is the equivalent of CDK, MCDK in vertebrates. This is in fission yeast. And the residue was later identified to be Y15, so tyrosine 15 residue was identified to be the residue in which V1 transfers this phosphorylation group. The next question was, which of the following chemicals would lead to suicidal mitosis? So there are a few options over here, and this can be explained with this experiment in which they observed that in control cells there is normal mitosis. However, if you add caffeine to the cells, visibly there is no change and the cells undergo normal mitosis. But what actually is happening is that caffeine is inhibiting a cell cycle checkpoint. Because of this, even if there is some problem in the S phase of the DNA replication in these cells, the cell will still undergo mitosis, but this is not visible in these cells. However, when we add hydroxyurea, there is a halt in S phase. This is because there is a decline in the production of DNTPs that is required for DNA replication, and there is a fork stalling. The cells have a fork, as in it is undergoing DNA replication. However, there is a lack of DNTP, so the cell cannot replicate the DNA. Hence, there is a fork stalling. and the cell cycle is halted. This is because of the checkpoint assembly factors, which it happens in the mitotic phase, that basically controls whether the cell is having replicated DNA or not. And now when we add caffeine to this reaction, we see that even though the DNA is not completely replicated, the cell will still undergo mitosis because the checkpoint assembly is now inhibited. And this is termed as suicidal mitosis because these daughter cells, they do not have sufficient amount of DNA in them and they are often non-viable. Hence, this is called suicidal mitosis. So the answer would be caffeine and HU together. Any one of them will not affect the cell survival. The next question was, which of the following is the correct statement regarding ring E3 ligases? Now, ring E3 ligases are a family of ubiquitin E3 ligases, which basically transfers ubiquitin to the substrate. Now, there are two families. One is the ring family, one is the Hecht family. In the Hecht family, if you see, the E3 ligase first forms a complex with ubiquitin and then it transfers it to the substrate. However, in the ring family of E3 ligases, this basically just acts as an adapter molecule. It brings the ubiquitin and it brings the substrate close to each other so that the substrate is transferred from the E2 enzyme to the substrate directly. So this is the cycle of ubiquitin transfer to the substrate. So what happens is first there is an E1 enzyme that attaches ubiquitin to itself. This is then transferred to the E2 machinery. The E2 machinery then has to transfer it to the substrate or to the E3 and then the substrate. If it transfers it to the E3 and then the substrate, then it is a Hecht family. However, if the E2 directly transfers it to the substrate, it is a ring family. So the answer would be that ubiquitin is transferred from E2 ubiquitin ligase to the substrate directly without being transferred onto itself. The next question is which of the following parts of the yeast proteasome system unfolds the protein to enter inside the proteasome? So the options are fairly simple. Deubiquitinating enzyme is the enzyme basically present in the regulatory components, and this removes the ubiquitin from the protein and pushes the protein into the proteasomal machinery to get degraded. The AAA-ATPase subunit basically is an ATPase enzyme which removes the phosphate group from the ATP, and this releases out energy that it converts into a mechanical force. And what it basically does is that it pushes the protein inside the proteasome, and in this process, it unfolds the protein. The core particle is actually the catalytic subunit, which chops up the protein. So the answer to this as to what unfolds the protein would be the AAA-ATPase subunit of the proteasomal system. The next question is, which of the following statements are incorrect about DNA damage checkpoints? So like I mentioned, DNA damage checkpoints basically are present in the cell to ensure that there is no damage in replication, or there is no mutated sequences, or there are no faulty replication that is happening or stalled replication that is happening. So if you look at the options, DNA damage checkpoints are regulatory pathways that delay cell cycle progression in response to DNA damage. This is a correct statement, and we have to identify the incorrect statement. Proteins involved in DNA damage checkpoints have been reported to control the movement of DNA repair proteins to the site of damage and activate transcriptional responses. This is also true because they initiate DNA damage responses, DNA repair responses, and ultimately end up in repairing the DNA damage and then progressing to the next stage of the cell cycle. In case of substantial damage, the cell may opt for apoptosis at the cost of losing a cell. This is also correct because if there is so much damage that the DNA repair proteins cannot repair them, then the system will rather risk losing a cell rather than allowing the cell to survive and cause other catastrophic problems. So the answer would be none of the above because all of these are correct statements. The next question was which of the following reagents will not result in the activation of a cell cycle checkpoint? This is again a fairly simple question. Hydroxyurea will result in activation because it stalls the S phase. Methylmethane sulfonate is a DNA damage protein which will alkylate the bases and cause DNA repair. UV radiation, again, we know causes DNA repair, and hence it is again none of the above because all three of them will cause some problem or the other in the cell and will hence activate a cell cycle checkpoint. The next question was why does the cell cycle need to be regulated to avoid unchecked cell division leading to tumor formation? Yes. To maintain proper cellular homeostasis. Yes, otherwise homeostasis is basically like a basal state which is maintained, a healthy physiological state which is maintained, which ensures that the cell is able to perform its function. If it's terminally differentiated, then it needs to be terminally differentiated and perform the functions that it is supposed to synthesize the proteins that is required of it and not devote its energy to replicating itself. To avoid abnormal growth of tissues, again, very similar to the first option, tumor formation. So all of these are reasons as to why the cell cycle, in fact, needs to be regulated. Next question is, which of the following statements is correct about the cell cycle? So in Saccharomyces cerevisiae, which is a yeast, after reacting to a certain size at the start, the cell gets committed to entering the G2 phase and ultimately completing the cell cycle. This is not true because G2 phase is not the place where the cell gets committed. In mammalian cells, the restriction point in G1 is analogous to start. This is the right option. Temperature sensitive CDC mutants can bud at non-permissive temperature. It can bud at permissive temperature, not at the non-permissive temperature. And CDC28 in yeast cyclin is the yeast cyclin. This is wrong because it's the yeast CDK. So mammalian cells, the restriction point is analogous to start in yeast. The final question was xenopus proved to be critical for understanding the cell cycle because the xenopus egg is very large in size. Yes, we can often see it with our naked eye. The egg is easy to work with. Yes, because it has a large cytoplasm. It can easily be injected and you can manipulate its contents. The egg undergoes multiple rapid cell divisions following fertilization. Yes, it often does not have a G2 phase. The cells do not grow. It just rapidly multiplies to form a suitable sized embryo. So the answer is all of the above. So I think this brings us to the end of the previous year assignment questions. And now I thought I would discuss a few techniques that is required to study the cell cycle that have been in fact mentioned over and over in the cell content, in the week's content. So it would be better to know what it actually is and how we perform these techniques in the laboratory. So we've been reading about the cell cycle, right? And most of us, if we had biology in our 11th and 12th, we would have performed this experiment, which is the onion root tip staining experiment, wherein we stain the chromosomes and we see different stages of cell division. And we can appreciate that, oh, there is anaphase happening, chromosomes have reached the poles of the cell, or the chromosomes are arranged at the center of the cell, metaphase is going on, or the cell is not dividing, or the cell has two nuclei and is yet to perform cytokinesis. So this process is often dramatic and can be easily visualized. And this is what was first identified. They just knew that the cell was condensing its chromosomes all of a sudden, dividing it into two and dividing the cytoplasm into two daughter cells. However, much later, using this technique known as EDU or BRDO incorporation assay, the scientists discovered that there was this specific stage in the cell cycle wherein the DNA was replicated into two. So this is a microscopic image of different stages of these cells. The blue is the nucleus of the cell. It's stained with DAPI. And different stages of the cell accumulate EDU differently. What EDU is, it's basically analog for the U nucleotide. And because of that, only replicating cells will be able to incorporate it into its system. So if your cell is having the DNA replicated, then more of EDU will be incorporated and there is fluorescence seen. Again, in the middle and late S phase, you can see fluorescence. However, at the G1 and the G2 phase, there is no fluorescence detected, suggesting that there is no DNA replication happening in these stages. What we can also appreciate in this image is that the size of the nucleus from G1 to G2 is significantly larger, suggesting that the cell grows. And in fact, that is why it's called the growth phase. So basically, we've seen these stages of cell cycle, and we know that there is something unique to every stage that can be used in these techniques to identify the different stages of cell cycle. So basically, G naught, which is the metabolically inactive stage. G1 is a metabolically active stage, right? There is so much proteins being synthesized that this cell is getting ready for what is to happen later. The S phase is the replicative phase, so that is where the DNA is being synthesized. And the G2 phase, in fact, is the phase where DNA replication is done. So it will have double the amount of DNA that a normal cell has. So if, say, we use an RNA label, which is a dye that stains all RNAs present in the cell. So there is a greater amount of RNA in the cell, suggesting that there is a greater metabolism happening in these cells. We know that it is in the G1 phase. This is a dye, which is pyronin Y, which labels RNA. And if there is a lesser amount of RNA, then we know that it is metabolically inactive and hence in the G0 phase. If you use a DNA label instead, DNA label can be DAPI, PI, or these uridine analogs, BRDU, CLD, which is chlorine, and IDU, which is iodine, instead of bromine and BRDU. Here, what happens is in your replicative phase, you will slowly see that if your cell has, say, 1x DNA in normal condition, the S phase might have 1.5, 1.2, 1.3, anywhere in between. And in G2 phase, it will have 2x. So this also can be differentiated. And this is what scientists do in the lab to differentiate between different stages of cell cycle. So this was this paper that I could find, and to what I am presuming was the first paper that established this technique of BRDU staining to see whether the cell has completed its S phase or is in the S phase or not. This is the paper that came out in 1985, much later than the Johnson Rao experiment. And here, what they basically do is they use two dyes to label the DNA and to label synthesizing DNA. So what is BRDU? This is basically a 5-bromo 2-deoxyuridine that is capable of replacing thymidine in the DNA. And here it has a bromine group. This can also be replaced by triturated thymidine, which is a radioactive thymidine residue, and this can be assessed by autoradiography. However, autoradiography is often not preferred because of the complications that come along with radiation and its regulations. So what the scientists prefer is they use this BRDU and then they use anti-BRDU antibodies, which is only going to label those cells that have taken up BRDU inside. So the concept that we need to be clear about right now is that the cell has ATGCs in its DNA. When it's synthesizing new DNA, it requires more of these T residues to be incorporated within the DNA, within the new daughter strand of DNA. Now, when we supply BRDU externally, the cell does not differentiate between the thymidine residue and the BRDU residue and incorporates the BRDU residue instead into its daughter strands. This can later be labeled by an antibody that will help us identify if a new daughter strand has been formed in these cells or not. So the protocol that this paper first established was first to expose the cells to BRDU for 30 minutes. And then there is a strong acid treatment. This basically allows the strands to, whatever strands are present in the cell, it is denatured and the BRDU binds. And the BRDU antibody is able to bind to the BRDU. Then we add the antibody against PRDU. This is generally a fluorescent antibody, so we can detect it with immunofluorescent techniques using a confocal microscopy. Then you label all DNA that is present with PI. So whether it just replicated or it was always present, it will be labeled with PI. And then we see whether we can differentiate between the cell cycle or not. So what is the figure that was represented in this paper was this, which is the flow cytometry experiment. So to explain what a flow cytometry experiment basically is that you have a cell population and you have two fluorescent dyes. One is this immunofluorescent antibody, which in this case was green fluorescence. So we'll remember that all actively replicating cells, as in if the DNA is replicating in these cells, if it's in their S phase, they will be labeled green. And this PI dye, which is red in color, which is the red fluorescence here, and all DNA in all cells, irrespective of what stage it is in, will be labeled with PI. So here we can see that there is this very characteristic curve that we can see over here in the cell population. So the x-axis over here is the red fluorescence. The y-axis over here is the green fluorescence. So first, what should we differentiate? First, we can differentiate between the different levels of red fluorescence in the cell. Say your cell has only X amount of DNA, which is the normal amount of DNA present. Then the red fluorescence will be less, say around 200 as shown in this figure. However, if it's in its G2 phase, that is, it has completed replicating its DNA. So now there are 2x the amount of DNA, it will have a higher fluorescence. So now if it was 200, it would have become 400. If it was 300, it will now be 600. The fluorescence intensity of the PI dye, the red fluorescence here. And then is the green fluorescence. So if the cell did not replicate in this short 30-minute time that you have given the BRDU treatment, then there will be no green fluorescence seen. So there will be very low amount of green fluorescence. We can appreciate in the figure on the right as well that there is very low amount of green fluorescence in cells that are in the G1 or the G2 phase because they have not replicated their DNA. However, if your cell was in fact replicating its DNA, that is, if it was in its S phase, it will actively accumulate the green fluorescence. The BRDU would have been incorporated into the DNA, and hence it has a high intensity of fluorescence seen. So basically, in this curve that we see here, we can divide the cell populations into three. G1, the leftmost population, S, the center population, and G2, the rightmost population. Now, say you've given the 30-minute BRDU treatment, and you let it be for 2 hours, and then you're fixing the cells to see what has happened now. And then here we can see that the S phase cells that were actively replicating are done replicating now, and they have moved to the right, which means that they now have the 2 times content of DNA, and all of these cells are in the G2 phase. And here we can see that the G1 phase, after 4 hours, we can see that the G1 phase are also slowly moving into the S phase, having greater green fluorescence. And at a later time point, we can see that most of the cells have accumulated green fluorescence, and the basal population that was initially seen having very low green fluorescence is lost, suggesting that most of these cells have replicated at least once in this time period. So say you want to differentiate between an actively dividing cell and a G0 cell. Can you do it using this technique? In fact, you can do that as well, in which you expose the cells to BRDU for 24 hours. And then you go through the same treatment. You treat it with acid to denature the DNA. Then you allow the antibody to bind. Then you add another fluorescent antibody that labels BRDU. So now wherever there's BRDU, there is fluorescent. Label all DNA with PI. Now, in this 24 hours, if the cell has not even replicated once, then there will be no green fluorescence in these samples. And hence you can conclude that these cells have been never replicating in this time point that we have been observing them, and hence they are in the G0 phase of cell cycle. So this was a paper that first established this technique, like I mentioned, and from then there are lots of advances in the same. And one such recent paper that I found was this dual EDU and BRDU pulse chase, which was recently published. In fact, I published almost exactly 1 year ago. So let us discuss basics of what this paper proposed. So what was the problem with the BRDU staining? One was that BRDU, the antibody does not bind to BRDU unless you kill the cells, add strong acid, and then allow DNA to denature. So you will not be able to visualize live cells that way. So you needed some other technique. And that is when scientists discovered this analog of thymidine again, which is EDU, which has an ethinyl group. This group again is incorporated into the DNA in the place of thymidine residues. However, what happens is when you just add a copper and a fluorescent probe, this is able to bind to these EDU residues without having to denature the DNA, and you can directly image these cells. So this was a technique that was earlier identified. But even though this was there are still certain problems. It's that the BRDU DNA staining or the EDU DNA staining measures the S phase fraction. We know that these cells have gone into the S phase, but we do not know how long this S phase is. S phase duration is different from S phase fraction. We know the fraction of cells that have entered S phase, but we do not know what is the duration of these S phase. So why do you actually have to study cell cycle is that there are these drugs that are given often to cancer cells, and that is known to stop replication of these cells. So what happens is in order to evaluate the efficiency of these drugs, you need to see if it has an effect on the cell cycle or not. You need to see if the cell is still able to do the normal events of S phase or not. So here, in such cases, to quantify the efficiency of a dye, you need a number. You need a quantification as to see whether the S phase duration is reduced, increased, or what is exactly happening. So in such cases, S phase duration becomes very important. Additionally, there are some heterogeneous groups of cell in which the S phase varies from one cell to another because of some stress or something, and that needs to be evaluated. And this will also be very useful in, say, understanding the effect of other molecules. Say you're studying a new regulatory molecule, and you need to know whether this regulatory molecule is playing a role in the S phase or not. Such basic research. So you're mutating this molecule and you're seeing if there is any difference or not. And again, in such cases, the duration in which the cell is in the S phase is very important. Now, there's this other technique that is live cell imaging, wherein they just take a live cell and they visualize it under the microscopy to see whether the cell is actively dividing, how long is it taking to divide, and all that. So you could argue that that would give us the length of the cell cycle. However, what happens is every cell population, the G1 phase varies from one cell to another. So in this case, we will not be able to average out because we cannot image a lot of cells together. Hence, we'll not be able to average out over, say, 10,000 or 20,000 counts of cells, and will not give us an accurate value. So that is why there was a need, and this is the motivation behind the research that these authors performed. And they developed a system in which they used both the dyes together, EDU and BRDU. So, uh, to, to those who do not know what a pulse chase experiment is, what basically they do is they give a pulse, which is a treatment of short duration of any molecule of interest. In this case, they give a pulse of EDU. EDU, again, we need to remember that EDU and BRDU only stains those cells in which DNA replication is happening. So say you're pulsing the cells with EDU, this will start getting accumulated in cells only when the cell has crossed the G1 phase and is now entering the S phase. So even if you are giving this to a heterogeneous population of cell, which means that one cell is in the S phase, one is in the G1 phase, one is in the G2 phase. It will incorporate this dye only if the cell is in the S phase. So this is a pulse, which means, as I said, 30 minutes in which you're giving extracellular treatment of EDU, and then you're changing the media and the cells do not have EDU anymore. And then it's the chase, in which you basically see what is happening to those cells. You chase the pulse that you're given, and at different time points, you fix the cell and you image them. So here in the first T0, there is no chase is 0. In T2, there is a small chase of, say, the entire duration is 10 minutes, then it's a small chase of 2 minutes, 4 minutes, 6 minutes, and so on. So here they have represented this method, and at the end of the chase, they are giving a second pulse of BRDU. So the first pulse is EDU, second pulse is BRDU. This is the control in which T0, EDU, BRDU are given next to each other. And then consecutively, slowly, they are increasing the time point and they're giving the second pulse. What ultimately happens is, again, if your chase is as long as your S phase entirely, when you're giving the BRDU pulse, the cells will not be able to incorporate it into their system because now the cell is not replicating its DNA. Hence, these cells will have only an EDU stain and will not have a BRDU stain. But if the chase is smaller than the length of the S phase, these cells will have both the EDU and the BRDU stain on them. So how a flow cytometry image should look is that if there is no chase, then all the cell population will be dual labeled. It will have both EDU and BRDU. So red and green together will give you a yellow signal. If there is a 2-hour chase, then say this is the cell that was first pulsed. Now 2 hours have passed. So these cells are double labeled, but this is the end of S phase. So these cells have not taken up BRDU. And then it's a 4-hour chase where again, more cells have moved on to the G1 phase and they do not take up BRDU anymore. Ultimately, there will be such a point where there is no cell that is dual labeled. And that will in fact be the time taken for this first cell to reach here, that is G2. So the point where dual labeling is gone is the average amount of length a cell takes for its S phase. And this is in fact what these authors used to analyze. So this is again a typical flow cytometric graph. This is how to get the result. On the x-axis is the EDU, on the y-axis is the BRDU. So if it is further to the right, then it means that the cells have taken up EDU. If it is further to the top, it means that the cells have taken up BRDU. Hence, this quadrant 2, are those cells that are double positive, taken up both EDU and taken up both BRDU. So if you see in the initial stages, 30-minute pulse, then there are double population. 2-hour pulse, still double population. 4-hour pulse, still there are population with double dye. 6-hour, 8-hour, 10-hour. And this is when we can see that the amount of cells having both the dyes have significantly reduced, suggesting that this somewhere in between 8-hour and 10-hour is the average length of the cell cycle in these cell population study. So the cell count in such techniques in flow cytometry, it can start from say 10,000 and go on to 40, 50,000 counts as well. And hence the accuracy that you get from flow cytometry is very, very high, and that you wouldn't have gotten with video microscopy. So this I found to be a very interesting technique that I thought that should be shared. Now, moving on, there are other techniques as well, often a huge population study in which there is this population of cell that is added to these microfluidics chamber. There's this heterogeneous cell population, and now you stain them with DAPI, which is a simple dye that stains DNA content, and then you calculate the percentage of cells that have say X amount of DNA and the percentage of cells that have 2X the amount of DNA. This will again not give you the S phase duration, but it will give you a average estimate of what fraction of your population is in the G2 phase and what fraction of your population is in the G1 or the S phase. And this is also a population study. And if your question is your question is to ask whether you want to identify the population dynamics of your cell, then this is a technique that can be used. Now, again, another technique that has been mentioned over and over in this cell cycle content is the temperature sensitive mutants. So this was a type of conditional mutant that was identified in yeasts. And I will just briefly explain as to how they are isolated and why exactly do they show these phenotypes. So I'm sure that you would have heard of this technique called replica plating, in which what you basically do is you have a set of colonies on a master plate. So say this is the master plate. You have a set of yeast colonies that you have subjected to DNA damaging agents. You've added EMS, MS, you've given UV radiation. And now there are certain mutations that have been incorporated into these colonies. So what you do is you take these colonies and you press them onto this velveteen block, which is a wooden block essentially. So now there are a few yeast cells that have been imprinted on this velveteen layer, and this can then be pressed onto another plate. Now you will have two identical plates. Exactly the colonies that were here are in this plate in the exact same location. So now you can give them two different conditions and compare as to is there a difference in their growth. Here they have the condition that they've used is complete medium and minimal medium. But in our case, if we want to isolate temperature sensitive mutants, then what you can essentially do is grow one plate in 37 degree, one plate in 22 degree, and see if there is a difference in growth. And then you can see that, oh, okay, this colony is able to grow at this temperature, but it is not able to grow at this temperature, meaning that there is certain temperature sensitivity that we can exploit. Now, this is what the initial researchers did in order to identify the regulators of cell cycle, and this is how CDCs were initially identified in yeast. What essentially happens is if it's in a permissive temperature, this polypeptide, whatever protein that has undergone some mutation, is able to fold properly, or there are certain chaperones that are active and helping in its folding, and it is active. And now if it's active, then the cell will behave as a wild type only. But at restrictive temperature, there is unfolding or misfolding. Unfolding in the sense it remains unfolded, because of which it is degraded by the proteasomal machinery, and there is a low amount of active protein. Or there is misfolding, in which it can form aggregates with each other and form like clumps and accumulate, again decreasing the amount of active protein. Or it can be stable but still have low affinity for the partner that it needs to bind to, and hence not express its catalytic activity. And so there is this phenotype that is observed. This is how the yeast CDCs were initially identified. Now, moving on, most of you in the last discussion session told me that you want to discuss certain recent papers and advances in this field. And to me, if I think of cell cycle, then definitely there is this one paper that changed the face of stem cells and how cell cycles were regulated, and deservingly was awarded the Nobel Prize in Physiology and Medicine in 2012. This was a study done by Professor Yamanaka, and basically established a new field of research. And so I thought that even though most of you must have heard of Yamanaka and Yamanaka's factors at one point of time or the other in your biology career, I thought it would be interesting to discuss the actual figures in the paper and to discuss as to how he actually constructed this experiment. So This paper was published in the year 2006 in the Cell Journal. It was titled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Culture by Defined Factors. So let's start with a little background as to what are stem cells and why are they so important. So stem cells are basically those cells that are able to divide and renew itself. So there are these two very essential properties of a stem cell. One is that it's able to divide, which means that it is able to undergo cell cycle and cell division. And two is that one of these daughter cells becomes its own phenotype and the other goes through a series of differentiation and can become other types of cells. So initially, this stem cells was first established when there were scientists who took a fertilized egg and identified that these cells were capable of forming the entire organism, including the embryo and placenta. These are called totipotent. Totipotent meaning they can form the entire population of cells that are present in your body. So, if you can think about it, your cells basically all arise from a single fertilized egg, but they are so different, and neurons are so different, and they have these dendrites. And on the other hand, RBCs are so different, they don't even have a nucleus, and they are small circular cells, but they all have the same DNA. This is because of these differentiation factors that are present, and this fertilized egg hence form totipotent stem cells. However, if you let them divide for a little bit more time, they form this embryo. And if you take the cells from the inner cell mass, they form cultured pluripotent stem cells. Pluripotent meaning they can form many different types of cells, yet cannot form the entire body, suggesting that we cannot form the placenta of the organism. So this was one of the first reports in which scientists first understood that these stem cells can be taken from these embryos and can be used to treat, say, various diseases. You take these stem cells from the embryo of an organism and add the specific factors that makes them neurons, they become neurons. Add specific factors that make them a muscle cell, they become muscle cells. And say somebody has a problem with their liver in the later stage, you could have taken this embryonic cells, made them into liver cells, and then transplanted that inside the body. So this was an unchecked, it had great medical potential, and hence there was a lot of research going on in this field. But there was this other very crucial experiment that Yamanaka says was very crucial for him deciding to progress with this experiment. This was that there was this scientist called as John Gurdon who took a fully differentiated somatic cell, which means that they have been terminally differentiated. They do not retain the cell cycle potential anymore. They are in the G0 phase. They are not going to divide. They're not going to replicate their DNA. But if they take such cells, take just the nucleus from this cells, take a unfertilized egg from a frog, the xenopus egg that we're talking about, remove the nucleus from this and add this nucleus instead. You see that this nucleus transplantation is enough for the normal embryo formation and a normal development of an adult frog, suggesting that the somatic cell also can, the nucleus basically containing the same DNA, same level of regulation, can be triggered to form an embryonic stem cell like nature just because of the contents that are present in the cytoplasm of these cells. So, moving on, what are induced pluripotent stem cells? What this paper basically proposes is that you have a patient and you can take any somatic cell from the patient, suggesting that you can take a section of their skin, take a section of their embryonic kidney fibroblast, kidney epithelial cells, and you add certain factors to them, and these somatic terminally differentiated cells somehow regain their property of being pluripotent and form these stem cell like cells that can then give rise to a large number of organs. So what this basically suggests that you do not need to preserve your embryonic stem cells. Say that you have not preserved your embryonic stem cells, but later on in the future you realize that there are certain problems because of which you have to replace, say, your muscle cells, and now you do not have your embryonic stem cells. Now you don't need it anymore. You just need a section of your somatic cells, and you can make IPS from them, which is in induced pluripotent stem cells, and then regenerate your muscle cells. So this experiment was in fact designed very beautifully. And for that, he used this gene cassette called as the reporter system. This is commonly used in day-to-day research, in which what they basically do is they take a promoter that is active only in certain stages. Say there is this promoter that is active only in a neuron cell. You add a GFP construct behind it. So this GFP construct will not be expressed anywhere in the body, but in the neurons, it will be expressed because it is only there that the promoter is active. So similarly, Yamanaka discovered that there was this promoter that was active only in the embryonic cell. So only if the cell has an embryonic stem cell-like nature, this promoter will be active. And then he cloned a reporter underneath. And this was a drug resistant marker. So say he used this drug known as G4128, I think, which is a neomycin like antibiotic. And he says that only if this promoter is active, as in only if the cells have become an embryonic stem cell like cell, the cell will express the resistant marker and hence it will not die. Otherwise, if the promoter is not active, the cells will die when you're giving it its drug. So let's focus here on the scenario 3 and the scenario 4. Say this construct is cloned into the coding region of the DNA, but the promoter is not active because the cell is not behaving like an embryonic stem cell, because of which you give the drug, the cell will not survive. However, if the cell has now regained an embryonic stem cell like nature, this promoter will be active, because of which this gene will be encoded and the cells will survive. So this is what he used, and he identified 24 gene candidates, which were proteins that were expressed in the embryonic stem cell, but not in any later stage of the life cycle. And he observed that if he added one factor at a time to these cells and then treated them with the drug, these cells all have the construct. So the drug is G418, and he observed that none of the cells survived, suggesting that none of these factors on its own can induce a stem cell like nature. However, a very interesting result, and this is the figure from the paper itself. Here we can see that there was this one well in which all the 24 factors were added together, and he could see that the cells survived, whereas in control cells, there was no cell survival. So again, this is him explaining the construct that he used. There is this promoter known as FBX15 that is only active in the embryonic stem cells. Now, if you clone a resistant marker, which is a neomycin resistant gene, and then you add neomycin to it, if the gene is active and it is being transcribed and translated, the cells will survive. However, if this is not active, then the cells will die. So then once he observed that adding 24 factors together, you see cell survival, you know that one of these 24 factors, or maybe a few in combination, is what is responsible for inducing this stem cell like nature. So then what he did was he took 24 gene candidates and added 23 of them. And then in each one of the well, he removed one factor. So say in well 1, he removed factor A. In well 2, he removed factor B, and so on. And every well had 23 factors minus 1. So in such cases, they saw that in certain wells, the cells did not survive. But in most of the wells, the cells did survive. Again, let's look at the figure from the paper in itself. Here we can see that on say adding factor 14 on depleting factor 14 or depleting factor 15 or depleting factor 20, there was no resistant colony observed at all, suggesting that these factors are very crucial to initiate this embryonic stem cell like nature. So next he short listed 10 of these factors and added 10 factors minus 1. Again, we see factor 14, factor 15, and factor 20 are very, very important. So then he shortlisted these factors into 4. And then he observed that upon adding 3 factors, that is 4 minus 1, it is still like even if colonies form, they're either very low in number or colonies do not form at all. And he proposed that these 4 factors is what is responsible for inducing a normal somatic cell into a stem cell like nature that can be used in medical applications. So here we can see that this is a embryonic fibroblast that he used, and here we can see that upon adding these factors, the cells regain a more circular, more rounded nature, and they behave more like embryonic stem cells instead. So these 4 factors were shortlisted to be OCT3-4, SOX2, CMYC, and KLF4. So in OCT3-4 and KLF4, it was fairly simple. There are no resistant colonies at all. But in SOX2, there were a few resistant colonies. But in CMYC, the colonies that were formed were having a non-embryonic cell-like phenotype. So here we can see that these cells have more rough edges, and even though these cells do survive, they do not seem like the IPS that were observed when all 4 of them were added. Hence, he concluded that all 4 of these factors are essential, and these are the only 4 factors that are required to make a somatic cell into a stem cell. These factors are today known as Yamanaka's factors. Now, there were a series of very interesting experiments that he carried out to see if such techniques had potential. And herein we can see that when embryonic stem cells were first isolated and then injected into a nude mice. So a nude mice is basically mice that lacks an immune response, and because of which any cell that you inject into them often develops into a tumor, and this tumor is known as a teratoma. So if you inject embryonic cells into this nude mice, you see a formation of a teratoma. Similarly, if you take somatic cells that are reprogrammed with Yamanaka's factor and you're inducing them to become pluripotent stem cells and you're injecting this into a nude mice, you can still see that there is a formation of a tumor. So both pathways lead to the same outcome, and hence this does act like an embryonic stem cell. This is, you can appreciate in the paper that these teratomas formed in fact had cartilages like cells, CNS, central nervous system like cell, muscle like cell, adipose cells, fat accumulating cells, and even epithelial cells, suggesting that this one induced pluripotent stem cell that he injected into a mice could now form various different tissues and hence could possibly even give rise to a completely new organism. There was this one other experiment that he performed that I thought was very interesting, was that he took a host embryo and then he inserted a few induced pluripotent stem cells into this and then transplanted this into the uterus of a mice. Now, when the organism developed, he could see that iPS cells that he injected were present in various organs of the adult mice. Now, these IPS he tagged with GFP, so only where these cells are present, it will have fluorescence and it can be detected with an anti-GFP antibody as well. So we can see that this was present in various organs of the mice, suggesting that the IPS indeed had a pluripotent capacity and could give rise to various different organs. And this is again the image from the paper in which we can see that this is the heart tissue. Here, the ones stained brown are his IPS, which have been tagged with GFP. So this is the gastrointestinal tract. Again, there are a lot of IPS present. The liver, the gonads, the neural tubes, and the skins had these induced pluripotent stem cells, suggesting that a chimera had indeed developed, and that iPS have uh, indeed the potential to give rise to different organs and different types of cells. Why I chose to discuss this paper is that this is a landmark paper that came out recently, and while most syllabus does cover this, what happens is they do not discuss the paper in itself and often cannot appreciate the results that he showed. Again, this is an example in which a cell cycle regulation has been explored in translational research, and actually a cell being able to divide is what has given this previously unknown potential to medical therapies. So that is all what I would like to say. Now we will discuss, and if you have any questions with what I discussed today or with what was in the course content, you can ask me.
Can you hear me?
Hello?
Hello, can you
Sorry?
hear me?
Yeah.
Yeah, this is Aishani. I was just wondering if there's any way of accessing the recordings of these sessions.
Yes. So I have recorded this session. This will
Right.
be this will be uploaded in the NPTEL portal itself. This will also be uploaded on YouTube. So
All
you can
right.
access it from there.
All right. So in the portal, where can I see it from? Do you have any idea?
So I don't think it has been uploaded yet, but what was proposed is either they will provide a link in the discussion forum or the announcement section, or they will just add another video.
All
But
right.
I think they will send a link to either all of you in mail or on the announcement portal on the SWAYAM
All right.
website.
So the one for week 1 has not been uploaded yet, right?
No, no.
All right. Thank you so much. Because I missed that due to some unavoidable circumstances, so I wanted to catch up.
Okay.
And
They will.
another question was the previous year assignments. Is there any way for us to access them on our own?
I don't think so.
All right. All right. Thank you so much.
So Amardeep had said, Amardeep, is he there in the group? I don't think so, but he said, please explain the previous slide again. I am not sure at what point. So if any of you have any doubts, you can we can discuss that slide again.
Good evening, ma'am.
Hi.
Ma'am, like you explained about one slide about the promoter and for the like IPS section in the like you explained in one slide like how he like took the promoter and the antibiotic resistant genes and like
Yes.
like why what was the reason he took that? I mean like I'm not familiar with those techniques. Like can you just quickly explain one more time? Like
Okay. So this is known as a reporter assay. So basically, like I said, every cell of yours has the same DNA, right? Like your neuron cells have the same DNA as your muscle cells, but they express different proteins, which is what gives them their unique nature. So there are certain promoters as in the regions that is present in front of a DNA segment that is encoding an mRNA that will later encode a protein. Now there are these regions that will specifically be only expressed in certain cell types. So what he wanted to know is he wanted to study what cells have been transformed into embryonic stem cells. So he chose a promoter. I will share that slide again. He basically chose a promoter called the FBX15. This promoter is only active in embryonic cells. So whatever you encode below that or under that promoter will only be expressed in cells that are embryonic stem cells in nature. So if I added a GFP over here, only embryonic stem cells will have that GFP fluorescence. What he did instead was he added a resistant marker. So now if you give normal cells this antibiotic, they will not be expressing the resistant marker and hence will die. However, if you are express or adding this antibiotic to an embryonic stem cell, this has an active transcription of this locus, because of which this antibiotic resistance will be translated and this protein will be available. So the antibiotic will not be able to affect the cell and hence the cell will survive. So what he basically wanted to see was whether this cell is being able to go back to its embryonic stem cell like nature. So because of that, only those cells that have gone back and have become stem cell like cells, or what he later termed as iPSCs, will be having the resistance gene. Others will not show resistance and hence will die. That is how he saw that if you add all 24 factors together at once, the cells are surviving, meaning that the cells have become stem cells.